Desmosomes are intercellular adhesions that help tissues, such as the skin and heart, to resist mechanical stress. The adhesions are formed by two types of adhesive cadherin receptors, desmogleins and desmocollins, which span the plasma membrane and are connected via a group of cytoplasmic plaque proteins to the intermediate filament cytoskeleton. Kathleen Green from Northwestern University Feinberg School of Medicine has a long-standing interest in how desmogleins and desmocollins assemble into desmosomes.
To me, it's always been fascinating to try to understand how those cadherins are synthesized and then targeted to the plasma membrane to their final site. And this project was one of the first steps to really look in detail at the molecular machinery that's responsible for getting the cadherins to their final place.
Green and colleagues, led by postdoc Oksana Nekrasova, found that the desmosomal cadherins desmoglein 2 and desmocollin 2 localize to distinct cytoplasmic vesicles in epithelial cells, but, as suggested by previous reports, both of these vesicle populations move to the plasma membrane along microtubules.
Fluorescently labeled desmogleins and desmocollins physically move along labeled microtubules in living cells. And using the cortisol to deplete the cells of microtubules, we can show that the appearance of the desmosomal cadherins is impaired at the membrane.
Nekrasova et al. Suspected that plus-N-directed kinesin motors might move desmosomal cadherins along microtubules. In collaboration with Jerry Kreitzer's lab at Weill Cornell Medical College, the researchers initially looked at the potential role of kinesin 1 in this process.
We started out with confocal analysis to look at the potential colocalization of kinesin heavy chains in the kinesin 1 motor with desmoglein 2 or desmocollin 2, and did see evidence that there was more localization with desmoglein 2. But it wasn't that satisfying, so we ended up using something called proximity ligation assay to really clean up the staining so that we really could see more clearly that desmoglein 2 was associated with kinesin heavy chain in situ, but not desmocollin 2.
The researchers also found a biochemical interaction between the heavy chain of kinesin 1 and the cytoplasmic tail of desmoglein 2. This is likely to be an indirect association, but it appears to be specific, as no interaction was seen between kinesin 1 and desmocollin 2.
We again used live cell imaging and could also show specificity in this way. Long-range movements of desmoglein 2 were really efficiently blocked by specifically knocking down the kinesin heavy chain or using a dominant negative kinesin 1.
Desmoglein 2 transport could be rescued by re-expressing the kinesin 1 heavy chain in RNAi-treated cells, but in the absence of kinesin 1 function, desmoglein 2 failed to accumulate in desmosomes at the cell surface. These desmosomes still contain desmocollin 2, as well as cytoplasmic plaque proteins like plakoglobin and desmoplakin.
So once we saw the kinesin 1 dependence of the desmoglein transport, but desmocollin didn't appear to be affected, we were left then with, well, how is desmocollin getting to the plasma membrane?
The researchers turned their attention to another member of the kinesin family, kinesin 2.
We used a similar approach where we knocked down either the heavy chain, which is called KIF3A, or this kinesin-associated protein, which is part of the kinesin 2 motor. It's called CAP3. And all of these impairments of the kinesin 2 motor affected desmocollin 2 trafficking and accumulation of the plasma membrane, but apparently not desmoglein 2. So again, there was a specificity there for the kinesin 2 desmocollin transport.
Desmocollin 2 also showed a specific interaction with kinesin 2. The desmosomal plaque protein plakophilin 2 may be involved in this association because depletion of this protein also inhibited desmocollin 2 transport but not desmoglein 2 movement. The two types of desmosomal cadherins therefore move independently to the plasma membrane, and cells deficient in kinesin 1 or kinesin 2 assemble desmosomes lacking desmoglein 2 or desmocollin 2 respectively. So what effect does this have on intercellular adhesion?
We lift up a confluent sheet of cells that are adhering to each other and then subjected to mechanical stress. And that mechanical stress will lead to a number of fragments if the sheets aren't sticking together very well, or the sheet may maintain its integrity. So we were also able to use that assay and showed very specific effects of knocking down the individual kinesins on desmosomal cadherin-mediated adhesions.
Knocking down either kinesin increased the fragility of cell monolayers, indicating a reduction in adhesion strength. Tissue integrity wasn't compromised further if the corresponding cadherins were also knocked down, but the simultaneous inhibition of both types of desmosomal cadherins made the monolayers much more fragile.
So we thought that was also supporting data for the specificity of the motors for their cadherin molecules.
So why are different desmosomal cadherins transported to the cell surface by distinct kinesins? One possibility, Green says, is that this may allow cells to modulate their adhesive properties.
I think there are a lot of implications for being able to tailor adhesion during development and then during pathogenesis. For instance, during cancer or even wound healing, where you may want opportunities to regulate adhesive strength. I could imagine that the targeting of desmosomal cadherins to the membrane, one of them may, for instance, be important for nucleation of desmosomes, but strong adhesion may not occur until the other one gets there and you get a complete clustering into a final desmosome. This is pure speculation, but I think the opportunity to regulate adhesion in different ways is there because of the different motor proteins that are trafficking the cadherins to their final resting spot.
Because mutations in desmosomal proteins are associated with a variety of skin and heart diseases, Green and colleagues are also interested in determining whether defective trafficking underlies any of these disorders.
One disease in particular that we're interested in, because mutations in the tail of desmoglein 2 have been identified in this disease, is something called arrhythmogenic right ventricular cardiomyopathy. We think it's possible that this could affect protein interactions, which affect the junction. Some of those protein interactions possibly could be important in trafficking. This is the direction that Oksana, who did a super job with this project, is going to be testing.
In the meantime, you can read more about the transport of desmosomal cadherins by distinct kinesin motors in the paper by Nekrasova et al., published in the December 26, 2011 issue of the Journal of Cell Biology.